Hello friends, I am Poonam, biology teacher. Today my topic of presentation is sexual reproduction in flowering plants. We all know that flowers are the site of sexual reproduction. All flowering plants which are known as angiosperms show sexual reproduction. There are some pre-fertilization structures and events occurs in the plants. Several hormonal and structural changes result in differentiation and development of the floral primordium. Development of flower occur by some hormonal changes, and vegetative bud convert into reproductive bud and finally flower formed. Now about the endoecium. Endoecium is the male part of the flower. Today we will discuss about the male part of the flower. This is the flower. We can see here both structures, female reproductive structure and male. Today our topic is male reproductive structure. This we can see here, this is the male reproductive part of the flower, which is known as the anther. Collectively, if we know that endoecium is made up of the stamens. Okay, now parts of the stamen. Stamen is basically made up of two parts. One is known as the anther and another one is the filament. We can see in this picture that this long slender stalk is known as the filament. The proximal part of the filament is attached to the thalamus of the flower. Okay, now about the anther. Anther is the terminal parts of the stamen and it is bilobed. It has two theca and the thecas are separated by a longitudinal groove. Now we will discuss about the transverse section of the anther. The anther is a tetragonal structure consisting of a microsporangia located at the corners. And the microsporangia develop into the pollen sacs and these pollen sacs are filled with the pollen grains. Okay, now about the structure of the microsporangia. This is the structure of the microsporangia. Generally we can see that the microsporangium is generally in circular in outline. It is surrounded by four walls. Epidermis, the outermost layer, then the endothecium inner to the epidermis, middle layers, then the innermost layer is known as the tapetum. The outer three layers, epidermis, endodermis and middle layers function is to protection to the microsporangium and it also helps in the dehiscence of the microsporangia. Okay, now about the microsporangium. We have already discussed about the four layers of the microsporangia. We will discuss here in this slide only about the tapetum. Tapetum is the innermost layer of the microsporangia which provide nourishment to the pollen grains. And also cells of the tapetum has dense cytoplasm and it have also more than one nucleus. In young anthers, each microsporangium has sporogenous tissue which is made up of the sporogenous cells. Now about the microsporogenesis. Microsporogenesis is the process in which male gametes are formed. In this diagram we can see that microspore mother cells. In this microspore mother cells, meiosis 1 occurs and there is the formation of the microspore dyad. After meiosis 2, there is the formation of the microspore tetrad and these ultimately forms the pollen grains. Each microsporangium contains thousands of pollen grains. They are released with the dehiscence of anther. Now the pollen grains. You can see here the variety of the pollen grains. Maximum the pollen grains are spherical in shape. The diameter of the pollen grains are 25 to 50 micrometer. Cytoplasm is surrounded by a plasma membrane and pollen grains has two walls. Outer one is exine and inner one is antine. Now we will study about the exine. This is the structure of the pollen grains. You can see the outermost layer, it is known as the exine. Somewhere you find that somewhere the exine is absent. These are known as the germ pores. And this exine is made up of the sporopollenin. This sporopollenin is the substance which is very highly resistant to organic substances. Now about the intine. The intine is the inner wall of the pollen grain and it is made up of cellulase and pectin. Now the mature pollen grains. This is the structure of the mature pollen grain. Pollen grain divides and it forms two cells, vegetative cell and the generative cell. The larger one cell is known as the vegetative cell and the smaller one is known as the generative cell. You can see that the generative cell lies in the cytoplasm of the vegetative cell. Now the pollen grain. About the pollen grain, over 60% of angiosperms, the pollen grain shed off at the stage of two cell stage. One is vegetative and another is generative cell. Thus pollen grains are in some of the 40% angiosperms, they can shed in the stage of the three cell stage. One is vegetative and two male gametes are there. Now about the viability of the pollen grains. The shed pollen grains have to land on the stigma before they lose viability. This we can see the female structure, female reproductive part of the flower, and this is the pollen grain. The pollen grains will have to shed on the surface of the stigma for the fertilization. Because the viability period for the pollen grain is different in different types of crops. It can be vary from the 30 minutes to many months. In case of Solanaceae family, Rosaceae families, the viability period of the pollen grains are up to several months. So it will have to shed on the stigma. Thank you. It was all about the male gametophyte. Now in next presentation, I will take the female reproductive part of the flower.